Okay, welcome to the channel. Are you ready to test your knowledge in biochemistry about proteins and amino acids? So we have 30 exam style questions and you'll be given 5 seconds to answer each question. Let's begin. Question 1. Which amino acid is classified as an essential amino acid? Which amino acid is classified as an essential amino acid? A. Alanine, B. Glycine, C. Leucine, D. Serine. You have 5 seconds. Okay, so the answer is C. Leucine. So leucine is an essential amino acid because the body cannot synthesize it. It must be obtained from the diet and plays an important role in protein synthesis, muscle growth, and also tissue repair. Why other options are wrong? So option A, alanine is a non-essential amino acid because it can be synthesized by the body through transformation reactions. Then option B, glycine is also a non-essential amino acid and is produced naturally in the body. It is important for collagen synthesis and neurotransmission. Then option D is also wrong because serine is a non-essential amino acid synthesized from glycolytic intermediates and is involved in many metabolic pathways. Okay, so here is an exam tip. A quick way to remember this is essential amino acids must be supplied by the diet because the body cannot make them. Okay, let's go to question 2. Which amino acid group has a positively charged side chain? Which amino acid group has a positively charged side chain? A. Basic, B. Acidic, C. Non-polar, D. Aromatic. You have 5 seconds. So the correct answer is A. Basic. So basic amino acids have side chains that become positively charged at physiological pH. So lysine, arginine, and histidine are the major basic amino acids and play important role in protein interactions. Okay, then why other options are wrong? So let's start with option B. Option B, acidic amino acid, they usually carry a negative charge because they contain an addition carboxyl group. Okay, then option C, non-polar amino acid have hydrophobic side chains and do not carry a positive charge. Okay, then option D, aromatic amino acids contain a ring structures that contribute to protein stability but are generally uncharged at physiological pH. Yeah, so here is an exam tip to always remember. So remember the 3 basic amino acids, which are lysine, arginine, and histidine. They are frequently tested in medical examinations. Okay, let's go to question 3. So question 3 reads, the primary structure of a protein refers to its primary structure of a protein refers to its A folding pattern, B amino acid sequence, C, three-dimensional shape. D, protein association. You have 5 seconds. Okay, so the correct answer is B, amino acid sequence. Okay, so the primary structure is the exact sequence of amino acids joined together by a peptide bond. So the sequence determines how the protein folds and ultimately performs its biological function. Okay, why other options are wrong? Option A, it refers to protein folding rather than the amino acid sequence and is associated with higher levels of protein structures. Okay, then option C is wrong because it describes the tertiary structure, which is the complete three-dimensional shape of a single polypeptide chain. Okay, then option D is also wrong because it refers to the association of multiple polypeptide chains, which is the quaternary structure. Okay, so here is an exam tip. Remember the order of protein structure from primary to quaternary. Okay, so primary equals amino acid sequence. Remember that. Secondary structure equals alpha and beta sheets. Remember that. Tertiary equals one complete three-dimension protein. Remember that. And also quaternary equals multiple protein chains. Okay, so that's that. Let's go to question 4. Which bond stabilizes the primary structure of proteins? Which bond stabilizes the primary structure of proteins? So A, hydrogen bond, peptide A, B, peptide bond, C, ionic bond, D, disulfide bond. You have 5 seconds. Okay, so the correct answer is B, peptide. So peptide bonds are strong covalent bonds that join amino acid together to form the primary structure of proteins. Okay, so they create the backbone of every protein molecule. Should always remember that about peptide bonds. Let's go. Why other options are wrong? So option A is wrong because hydrogen bonds stabilizes secondary structures such as alpha and beta sheets rather than the primary structure. Okay, option C is wrong because ionic bonds help stabilize the tertiary structure through interactions between charged side chains. Okay, then option D is also wrong because disulfide bonds strengthen tertiary and quaternary structures by linking cysteine residues together. Okay, let's go to the exam tip. So the exam tip is a useful memory aid is, so always remember, peptides builds the chain, hydrogen folds the chain, disulfide strengthens the chain. Let me go through that again. So peptide builds the chain, hydrogen folds the chain, disulfide bond strengthens the chain. Okay, let's go to question 5. So question 5, alpha and beta sheets represent which protein structure level? I think this one might be simple for you now because we have discussed it in the previous questions. So alpha and beta sheets represent which protein structure level? A, primary, B, secondary, C, tertiary, D, quaternary. So we have 5 seconds to answer this question. Okay, so the correct answer is B, secondary. So the secondary proteins, as we earlier discussed, it is a structure that consists mainly of alpha and beta pleated sheets. This structure are stabilized by hydrogen bonds, as we discussed, between peptide backbone atoms, and are essential for proper protein foldings. Okay, why other options are wrong? So option A, it refers only to the amino acid sequence and does not include folding patterns. Okay, then option C, it describes the complete three-dimensional shape of a single polypeptide chain formed after secondary structure fold further. Okay, then option D, it refers to the arrangement of two or more polypeptide chains into one functional protein. Okay, then the exam tip is, whenever you see alpha and beta sheet, immediately think of secondary structure. This is one of the highest yield fact in protein biochemistry. Okay, so for today we'll end here. This is the end of part 1, so look out for part 2, which starts from question 6 going to question 10. Thank you.